Structural biology is looking at life in 3D. All biological processes, when looked at on the microscopic level, involve an interplay between DNA, RNA, and proteins. To find the 3D structure of these biomolecules is to discover the relative orientation of their atoms, which in turn gives us an idea of how they function. At Purdue University, the Hochmeier Hall of Structural Biology is well equipped to conduct X-ray crystallography and cryo-electron microscopy, two key techniques that can reveal the atomic structure of biomolecules. Let's look at how we do it. It all starts with the identification of a target molecule. This is the BAM complex. It is an outer membrane protein complex consisting of 5 proteins, BAM A through E. This complex is present in all gram-negative bacteria, which include pathogens like Neisseria gonorrhea, which cause deadly disease. Discovering the 3D structure of the BAM complex can help us design drugs that can bind to it, block it, thereby killing the pathogen. Structure-based drug design is one of the many applications of structural biology. The first step in revealing the structure requires the DNA sequence of a protein. This is followed by molecular cloning, where we insert this piece of DNA into a bacterial E. coli cell. We then produce these cells in nutrient-rich media and induce them to produce the protein of interest. After harvesting the cells, we lyse them, cracking the cells open under high pressure to extract the protein they have produced. This lysate is then taken through many purification steps involving techniques like chromatography to finally acquire purified protein. The purity of this protein can be checked by running them on SDS Page gel. Each band you see here is a component of the BAM complex. Now that our protein sample is ready, we can do structural analysis on it. To reveal the structure using X-ray crystallography, one needs to first crystallize the protein. This is done by chemical screening and plates such as these. We use robots like the mosquito and the dragonfly to automate the chemical screening process. This crystal hotel stores these plates and images them at regular intervals to check if a condition becomes suitable for crystal growth. Once we identify a protein crystal, we loop it under a microscope and freeze it in liquid nitrogen, which is at a negative 320 degrees Fahrenheit. The crystal is then mounted in an X-ray machine like this. A rotating copper anode generates the X-rays, which are guided towards the crystal. Once they hit the crystals, the X-rays get scattered and are captured at the detector. The output is called a diffraction pattern that looks like this. Using computational tools, we can use this diffraction pattern to acquire the 3D structure of the protein. Another emerging technique for determining protein structures is cryo-electron microscopy, or cryo-EM for short. Here, we do not need to crystallize our protein of interest, but instead dispense our protein onto tiny grids. We can then either stain the grid for negative stain electron microscopy, or plunge freeze it using this instrument to prepare the sample for cryo-EM. This is the Titan Krios, a state-of-the-art electron microscope. Grids are mounted here after being frozen. The electron microscope then images these grids to acquire 2D views of our protein in all different orientations. Using computational software, we can use thousands of these images to reconstruct a 3-dimensional view of our protein of interest. Discovering the structure of a protein is just the beginning of many more exciting paths. This structural information can be used to create realistic movies called molecular dynamics simulations, and the protein can be mutated to determine the mechanism of action. Drugs can be designed to bind proteins and modify their function. These drugs can cure diseases, unlocking the full potential of medical applications. The future of structural biology looks bright, and the large number of Nobel Prizes awarded in this field is a testament to that fact.